NMS Microbiology, Chapter 1, Overview of Medical Microbiology. First, Introduction. A. Medical microbiology in the study of interactions between humans and the microorganisms with which they coexist. The nature of these interactions varies from beneficial to harmful, and the microorganisms involved in these interactions are classified within the context of this spectrum. First, commensals are organisms that routinely colonize body surfaces without doing harm, that is, the bacteria normally present in the gastrointestinal tract. 2. Pathogens are organisms that damage the human host either by direct invasion and injury, that is, Shigella species, or by the production of harmful toxic products, for instance, Clostridium species. B. The pathologic potential of an individual microorganism is often variable, influenced by both the intrinsic properties of the microorganism and also by the particular state of health of the human host. When normal host defenses are impaired, a normal innocuous commensal may become a lethal pathogen. Thus, the role of any microorganisms is dependent upon two sets of determinants. First, the term host defenses refers to a multifactorial system of protective mechanisms which function to prevent entry of microorganisms into normally sterile areas and which limit the spread of the invader after entry has been accomplished. A. These mechanisms provide barriers that may be weakened by a variety of insults, including direct physical trauma, systemic disease, drugs, and toxins. B. When the normal defenses are impaired, the individual loses the ability to combat infection and the injury caused by any potential pathogens, even those with low intrinsic virulence. In such cases, the compromised host often succumbs to infection. Second, virulence is the relative intrinsic ability of a microorganism to cause disease. Organisms of high virulence have evolved efficient mechanisms for circumventing normal host defenses. Virulent organisms are adept at gaining entry and readily do damage even when the inoculum size is small. C. The study of infectious diseases has had a dramatic impact upon human history, and much of the foundation of modern scientific medicine has been established as a direct result of the development of medical microbiology. In particular, the germ theory of diseases, that is, the recognition that a specific etiologic agent is responsible for a particular constellation of symptoms, is an outgrowth of the understanding of infectious disease. 10. The 19th century microbiologist Robert Koch formalized the concept of a specific microorganism is capable of causing human disease. Koch developed a set of methodologic principles, Koch's postulates, for establishing this etiologic role. These principles remain important guidelines in medical research. A. The microorganism should be present invariably in the diseased individual. B. The etiologic agent should be isolated from the diseased individual. C. Inoculation of the agent into a healthy experimental animal should produce a disease similar to that observed initially. D. The presumed pathogen should be isolated from the animal with experimentally induced disease. 2. These postulates have provided an important conceptual model for exploring the underlying pathology of many infectious diseases. It should be noted, however, that these conditions may not be met in circumstances in which the pathogen cannot be cultured in vitro or when the agent is not pathogenic for the available laboratory animals. Second, classification of pathogenic microorganisms. Pathogens vary in size and biologic complexity. Some are able to extract sufficient nutrients from an inanimate environment and hence may be cultured on an artificial medium. Others are incapable of growth outside the living host cells and are thus referred to as obligate intracellular parasites. Pathogens are divided into four major groups. A. Viruses are the smallest intact organisms which demonstrate pathogenic potential. Because of their size, they cannot be seen with a light microscope. First, viruses are obligate intracellular parasites that depend entirely upon the host cell's synthetic machinery for reproduction. Second, viruses contain only only one type of nucleic acid, either DNA or RNA, but not both. Third, after entering a host cell, the virus is first disassembled, thus releasing regulatory enzymes that take over the host cell's synthetic machinery. Under the direction of viral genes, the host cell diverts its activities toward producing new viral components, which are then assembled within the cell into new virions, complete virus particles. When these new virions are released, additional cells are infected and the cycle is repeated. B. Bacteria are both larger and more complex than viruses. Most bacteria are visible under the light microscope. Third, bacteria are termed prokaryotes because, unlike higher organisms, they lack true cell nucleus. Since no clearly defined nuclear membrane is present, the genetic material of the cell lies within the cytoplasm. 2. Unlike viruses, bacteria possess both DNA and RNA. Thus, they resemble the higher phyla. 3. Bacteria reproduce by binary fission. Many pathogenic bacteria are capable of independent growth and thus may be cultured on artificial media. Some bacteria, however, lack the ability to produce important metabolites and are thus obligate intracellular parasites. Among the latter group are the chlamydia and rickettsia species. These organisms must be grown on tissue culture layers if their recovery is necessary. C. Fungi are larger than bacteria and have a more advanced cell structure. First, as eukaryotic organisms, their genetic material is separated from the cell cytoplasm by a nuclear membrane. 2. Some fungi reproduce by budding yeasts, while others from growing colonies of attached organisms, molds. 3. Most pathogenic fungi exist in nature as environmental saprophytes, and human infection does not appear to be necessary for their life cycle. D. Parasite is a general term that is often used in a narrow sense to refer to a variety of protozoan and multicellular eukaryotic organisms that are capable of causing disease. Many parasites undergo complex life cycles that may involve several host species, including humans. The parasitic diseases remain major health problems and sources of economic drain, especially in underdeveloped nations. Third, clinical evaluation and treatment of patients with infectious disease. Clinical evaluation and treatment of patients with infectious disease. A. Clinical presentation. Signs and symptoms of infection may occur acutely or develop gradually. The particular clinical picture varies with the etiologic agent of the infection and with the state of health of the patient at the time of the infection. Many of the important signs of infection are relatively non-specific, and therefore workup must consider both infectious and non-infectious diseases as potential causes. Important clinical findings that raise the tissue of infection are any or all of the following. First, fever is perhaps the cardinal clue to the presence of infection. However, it is important to emphasize that not all infections cause fever and not all fevers result from infection. The pattern of temperature variation may sometimes offer a clue to the diagnosis. Non-infectious causes of fever include A. Neoplasms. B. Central nervous system, CNS disease. C. Rheumatic disease. D. Hypersensitivity reactions. E. Metabolic derangements. 2. Pain may be a reflection of inflammation and tissue destruction, or may occur as a result of the pressure on sensitive structures caused by an enlarging infected mass. 3. Weight loss may occur in the setting of a chronic infection. 4. Progressive debility is a vague but important symptom. It may be a reflection of the catabolic effects of infection, or it may be traced to more specific problems such as malabsorption, central nervous system disease, chronic meningitis, and cardiovascular impairment, endocarditis. 5. Changes in the physiologic function of a particular organ system, for instance, progressive shortness of breath as a result of pneumonia, alteration in mental status due to meningitis, and diarrhea caused by enteritis may reflect infection. 6. Abnormal laboratory tests results tests may suggest the presence of infection even in the asymptomatic patient. Common examples include abnormal chest X-rays, elevated sedimentation rates, elevated circulating leukocyte counts with abnormal differential cell counts, and the presence of white blood cells in normally acellular body fluids, for instance in the urine, cerebrospinal fluid, CSF, joint fluid, and peritoneal fluid. B. Diagnosis. First, the importance of accurate diagnosis. Diagnostic maneuvers are aimed at identifying the individual etiologic agent responsible for the particular clinical picture. Accurate diagnosis allows implementation of a specific treatment regimen, thus increasing the likelihood of a successful outcome. In the absence of an accurate diagnosis, the physician is often tempted to rely upon shotgun empiric therapies, which should be employed only as a last resort after all practical diagnostic modalities have been exhausted. Empiric antibiotic regimens often create problems due to a number of reasons, including the following. A. The empiric regimen chosen may miss treating a curable infection. B. Because attention is diverted from the need for further diagnostic studies, valuable time is wasted. C. The patient is exposed to the toxicities of potentially unnecessary drugs. D. Empiric antibiotics can confuse interpretation of diagnostic tests such as bacterial cultures. 2. Strategies for diagnosis. When the possibility of an infectious disease exists, several strategies may be employed to develop a diagnosis. A. An effort should be made to define the anatomic localization of the infectious process. The physician must determine which organ systems are actually involved, and diagnostic tests are planned to maximize the yield of information from these sites. B. The physician should consider which organisms representing each major phyla, for instance viruses, bacteria, fungi, and parasites, are likely to cause the particular clinical picture seen as a particular patient. Efforts to perform optimal cultures for the most likely organisms should be made. C. Orderly accumulation and interpretation of data from several sources must be employed. Since the medical history provides important information about infection, questioning should obtain the following data. First, exposure history. For instance, exposure to diseases in family and friends, to both domestic and wild animals, and to insect vectors, as well as history of travel, hobbies, and occupation. 2. A related health history, for instance, history of immunosuppressive illness and drugs, heart and lung disease, past trauma and surgeries, and past serious infections. Third, epidemiologic considerations, for instance, time of year that the infection was acquired and the history of similar problems in the community. 4. Social history, for instance, sexual activities and illicit drug use. 5. Reliable indices of infection, that is, documented fevers, shaking, chills, weight loss, and rashes. D. The physical examination may provide important information. Documentation of fever, weight, and blood pressure is needed. The patient should be examined carefully for localizing signs, in particular localized tenderness, heat, swelling, and erythema. Other important localizing agents should be sought as well, including stiff neck, heart murmurs, lung conditions, skin lesions, abdominal tenderness, bony tenderness, and adenopathy. The laboratory evaluation is directed by elements in the patient history and physical examination. First, material obtained from the suspected site of infection should be gram-stained and cultured. Specimens must be collected carefully and delivered to the laboratory in optimal condition. Culture results must always be interpreted in light of the normal microbiologic ecology of the sample site. The gram stain is especially important in evaluating specimens taken from a normally contaminated site. The presence of inflammatory cells and a predominant organism on a gram stain provides important confirmation confirmatory information of infection. Because antibiotic therapy can interfere with the isolation of an etiologic organism, every effort should be made to obtain all relevant culture material before antibiotics are started. 2. Biopsy and histologic examination of potentially infected tissues may be especially important in the interpretation of culture results when the specimens are obtained from normally unsterile sites. The pathologist should be informed of suspected etiologic agents so that appropriate tissue stains may be employed. 3. Serologic tests are especially important when the suspected etiologic agent is known to be difficult to isolate or identify. A variety of methodologies, that is complement fixation, immunodiffusion, radioimmunoassay, enzyme-linked immunoassay, counter-immunoelectrophoresis, and immunofluorescence may be employed, but the technique is not so important as an understanding of what is being detected. Antibody detection assays, such as those for Legionella immunofluorescent antibody, IFA, and Brucella agglutinins, as well as Mycoplasma complement fixation, detect an antibody response to the pathogen, and thus may be positive even in the absence of an active infection. Antigen detection assays, for instance, the cryptococcal antigen test and hepatitis B surface antigen test, detect components produced by the pathogen itself and are thus more reliable indications of active disease. 4. Skin testing. Delayed hypersensitivity testing may indicate past exposure to a pathogen, but rarely indicates the activity of a particular infection. 5. Radiologic testing, for instance X-rays and nuclear scans, may indicate the anatomic locus of infection, but rarely offers an unequivocal etiologic diagnosis. On the other hand, radiographically directed percutaneous needle aspiration of abscess cavities is becoming important both as a diagnostic and a therapeutic modality. Chapter 2, Basic Properties of Bacteria, Anatomy, Classification, and Physiology. First, Introduction. Many of the basic properties of bacteria are most easily understood within the framework of 3 fundamental characteristics. A. Bacterial structure. B. Bacterial classification. C. Bacterial physiology. 2. Bacterial structure. See Figure 2.1. A. Cell wall. A rigid cell wall provides protection and imparts shape to the bacterial cell. First, the principal structural component of the cell wall is a mixed polymer called the peptidoglycan. Figure 2.2. A. Peptidoglycan polymers consist of 2 hexose sugars, N-acetyl-glucosamine and N-acetylmuramic acid, linked to short peptides of identical chains of 4 amino acids. B. Peptidoglycan polymers contain several amino acids that are found only in bacterial cell walls. These acids are mesodiaminopimelic acid and the D-isomers of glutamic acid and alanine. 2. Bacteria are divided into gram-negative and gram-positive groups based on their cell wall structure. A. Gram-negative bacteria have thin peptidoglycan layers next to the cytoplasmic membrane with other major cell wall components exterior to the peptidoglycan. First, the outer layer or outer membrane of these bacteria is thicker than the peptidoglycan layer and is composed of lipoprotein and lipopolysaccharide LPS attached to the peptidoglycan. 2. The outer membrane consists of somatic O antigens and the endotoxin characteristic of gram-negative bacteria. B. Gram-positive bacteria have a heavy, rigid layer of peptidoglycan and teichoic acid, which is several times thicker than that of gram-negative cells. First, gram-positive cells are far more sensitive to the enzyme lysozyme, which hydrolyzes peptidoglycan by specific cleavage between the N-acetylmuramic acid and the N-acetylglycosamine of the glycan strand. 2. Penicillin specifically inhibits peptidoglycan synthesis. Therefore, gram-positive bacteria are generally far more sensitive to penicillin than are gram-negative bacteria. B. Capsules, flagella, and pili are found external to the cell wall. First, capsules are present on many bacteria, including several pathogenic species. A. Most bacterial capsules are composed of complex polysaccharides. B. Some bacteria produce polypeptide capsules composed of D-amino acids. C. Bacterial capsules lack the well-ordered structure found in the bacterial cell wall. 2. Flagella are present on many bacteria and are responsible for the motility demonstrated by these species. A. Bacterial flagella are composed of a single species of polypeptide and are driven by rotary action of a swivel-like basal hook. B. Some bacteria have many flagella distributed over their surface, peritrichous flagella. C. Some bacteria have a single flagellum or small bundles located at one end of the cell, polar flagella. 3. Many gram-negative bacteria have long, slender projections called common pili, fimbriae, which originate at the cell membrane and extend through the cell wall. A. The common pili are composed of a single protein type with molecules in the form of a helical filament. B. Common pili appear to play a role in bacterial adherence to surfaces. C. Sex pili are a less common type of pilus and appear to be specifically involved in bacterial conjugation. C. The cell membrane, a cytoplasmic membrane, is the actual barrier between the interior and exterior of the bacterial wall. First, removal of the cell wall leads to bacterial lysis because the cytoplasmic membrane is unable to withstand the osmotic pressures found in nature. A. Protoplasts are cells maintained in an osmotically protected environment following the complete removal of the cell wall. B. Spheroplasts are also osmotically fragile but result from the partial removal of the cell wall. 2. Cytoplasmic membrane exhibits well-defined selective permeability. 3. The bacterial electron transport system, the principal energy system, is located in the cytoplasmic membrane. 4. Mesosomes are complex invaginations of the cytoplasmic membrane seen in many but not all bacteria. Their function has not been fully elucidated. D. Cytoplasm contains only ribosomes and storage granules because the cytoplasmic membrane performs so many complex functions. First, ribosomes are complex structures composed of several RNA molecules and many proteins and are approximately 20 nanometers in diameter. Each ribosome is composed of two subunits with sedimentation coefficients of 50 Svedberg units S and 30 S respectively. B. Ribosomes function as the active centers for protein synthesis. C. The antibiotics streptomycin, tetracycline, and chloramphenicol all act by inhibiting protein synthesis on bacterial ribosomes. 2. Storage granules are not universally present in bacteria. Their presence varies with the type of bacterium and its metabolic activity. E. The bacterial cell lacks a nuclear membrane, that is, it is prokaryotic. First, the cellular DNA is concentrated in the cytoplasm as a nucleoid. A. Bacterial DNA consists of one long double-stranded circular molecule. B. Each nucleoid appears to consist of one DNA molecule. 2. A small percentage of the DNA in some bacteria persists of an extra chromosomal state as molecules called plasmids, which also are circular but are much smaller than nucleoids. Plasmids frequently carry genes involved in antibiotic resistance, R factor, matters. 3. Bacterial classification. Identification of bacteria is of enormous practical significance in clinical microbiology. A. Binomial nomenclature is applied to bacteria within a rigid framework of taxonomic rules. The most significant taxonomic subclasses are genus and species. B. Readily observable properties usually are the basis for bacterial classification and identification. First, Gram staining reflects the most important properties of microorganism for classification purposes. A. If bacteria take up and retain the primary dye, crystal violet, or resist alcohol decolorization, they are considered Gram-positive. B. If the crystal violet is completely removed by alcohol decolorization, the organisms are considered Gram-negative. C. These simple staining reactions reflect the basic structural difference in the cell wall between the two cell types. 2. Morphological features are the second most important factor in classification. A. Bacteria may exist as: first, rods, bacilli; second, spheres, cocci; third, spirals, vibrios, and spirilla. B. Bacteria are found singly or in regular groups of 2 or more. C. Bacteria may have flagella over their entire surface, peritrichous, flagella at one or both poles, polar or no flagella. C. Metabolic reactions form the precise basis for bacterial species identification. First, reaction to growth in the presence of oxygen, O2. A. Aerobic bacteria tolerate O2. Anaerobic bacteria cannot tolerate O2. 2. Utilization of carbohydrates. A. Oxidation requires elemental O2 to degrade glucose. B. In fermentation, glucose is degraded anaerobically. C. Some organisms fail to use glucose under any conditions. D. Species can be identified by the variety of carbohydrate substrates used as energy sources. 3. Production of detoxifying and oxidative enzymes. A. Catalase destroys hydrogen peroxide. B. Oxidase transfers hydrogen directly to O2. 4. Production of proteolytic or toxic enzymes. A. Hemolysis lyse red blood cells, RBCs. First, alpha hemolysin lyse but do not dissolve RBC membranes. 2. Beta hemolysins both lyse and dissolve red blood cell membranes. B. Coagulase causes clotting of blood plasma. C. Gelatin liquefaction provides evidence of proteolytic enzymes. D. Genetic relatedness is based on similarities of nucleic acid sequence and provides a more phylogenetic scheme of bacterial classification than do the other methods. This type of classification can be used on: first, DNA homology between bacteria. 2. Nucleotide-based composition, which provides a broader classification scheme. 3. Exchange of genetic information occurring between related organisms. A. Transformation. B. Conjugation. 4. Bacterial physiology. A. Bacterial nutrition. The proper environment for the optimal growth of bacteria provides a variety of nutritional requirements. First, the proper O2 tension is essential for balanced growth. A. The growth of aerobic bacteria in a rich nutrient medium is usually restricted by a limited availability of O2. B. The growth of anaerobic bacteria, on the other hand, may be inhibited by O2 tensions as low as 10 to the minus fifth atmospheres. 2. Many microorganisms require carbon dioxide, CO2, for growth, and many are stimulated by CO2 partial pressure, PCO2, levels greater than those normally found in air. 2. The inorganic ions phosphate, PO4, 3-, potassium, K+, magnesium, Mg2+, nitrogen, N, and sulfur, S2-, are as well as large number of trace metals are essential for bacterial growth. 4. A variety of organic nutrients are required for bacterial growth, with the requirements varying widely among different species. A. Carbohydrates are used as an energy source as well as the initial substrate for many biosynthetic enzymatic pathways. B. Many bacteria have an absolute requirement for amino acids and in some cases for short chain polypeptides. C. Vitamins, purines, and pyrimidines are also common nutrients and necessary for growth. B. Bacterial growth. Bacteria increases their cell mass and numbers in an exponential manner. First, the bacterial growth curve, figure 2.3, is generally separated into at least 4 distinct phases. A. An initial lag phase is a period of physiologic adjustment involving the induction of new enzymes and the establishment of a proper intracellular environment. B. A period of logarithmic growth is characterized by maximal rates of cell division and mass increase. C. The stationary phase occurs as the essential nutrients begin to disappear, and it consists of a balance between cell growth and division and cell death. D. The death phase is caused by bacterial lysis and cell destruction, resulting in a loss of cell number. 2. Bacterial growth is regulated by the nutritional environment as described as well by intracellular regulatory events, feedback regulation. A. End product inhibition occurs when the first enzyme in a pathway is inhibited by the end products of that pathway. B. Repression of enzyme synthesis through catabolite repression is another device for controlling the metabolic activity of a pathway. 3. The rate of bacterial growth also is dependent on temperature. Most bacteria can grow in a wide range of temperatures but have a narrow range for optimal growth. C. Bacterial energy metabolism. The major forms of bacterial energy production are fermentative, respiratory, and autotrophic metabolism. 1. Fermentative metabolism is a metabolic process in which organic compounds serve as both the electron donors and the electron acceptors. A. The major fermentation pathway used by bacteria is the glycolytic Embden-Meyerhof pathway, which converts glucose to pyruvate. 1 mole of glucose is converted to 2 mole of pyruvate. 2. Glycolysis results in a net yield of 2 mole of adenosine triphosphate ATP per mole of glucose fermented. B. Many microorganisms use pyruvate in secondary fermentation to act to oxidize the reduced form of nicotinamide adenine dinucleotide NADH produced during the glycolytic fermentation. First, the simplest secondary fermentation is lactic fermentation, which converts pyruvate to lactate. 2. In alcoholic fermentation, pyruvate is converted to CO2 and ethanol. 3. In propionic fermentation, pyruvate is converted to oxaloacetate with the addition of a CO2 molecule, and then eventually it is converted to propionic acid. 4. Mixed acid fermentation, common in most Enterobacteriaceae, is a combination of lactate, acetate, and formate production via several similar pathways. 5. Many secondary fermentations result in the production of gas, CO2, hydrogen, H2, or methane, CH4, during the breakdown of pyruvate. C. Many organisms use alternate pathways to metabolize glucose. First, the major hexose degrading pathway in pseudomonads is the Entner-Dudorov pathway. 2. Some organisms use both glycolysis and the hexose monophosphate shunt. 2. In respiratory metabolism, the electron acceptor is usually O2, whereas in fermentation, the electron acceptor is an organic molecule, A. In the most common form of respiration, pyruvate from glycolysis is converted to CO2 and acetyl coenzyme A, acetyl CoA. The acetate is further degraded via the tricarboxylic acid rapid TCA cycle in a process termed terminal respiration. First, complete respiration increases the energy yielded by 1 mole of glucose from 2 mole of ATP produced by fermentation to 38 mole of ATP. 2. Respiratory metabolism also permits the use of energy sources that are so highly reduced that they cannot be fermented. B. A number of bacteria lack the enzymes needed for complete oxidation and therefore obtain energy from incomplete oxidations. C. The electron movement in the respiration process is funneled into the electron transport system. First, electrons move through a series of reversible electron carriers such with successively higher redox potentials. 2. These electron acceptors are embedded in the bacterial membrane and span the range from NADH to O2. 3. In 3 stages of the electron transport sequence, energy is used to extrude a pair of protons H plus across an insulting membrane, thus creating an electrochemical gradient, proton motive force. 4. The return of the protons through the membrane reverses the activity of adenosine triphosphate ATPase and is termed oxidative phosphorylation. 5. Cytochromes, hemoproteins, flavoproteins, and ubiquinones, coenzyme Q, are the major elements of the electron transport system. 3. In autotrophic metabolism, bacteria use a variety of alternate inorganic sources for energy and reducing power. A. In photosynthesis, bacteria use energy from light to convert CO2 to triose phosphate and subsequently to other cell constituents. B. In anaerobic respiration, bacteria use inorganic substrates as terminal electron acceptors in place of O2. D. Biosynthetic pathways. The enormous diversity in the nutritional requirements of bacteria stems from the biosynthetic abilities of different groups of bacteria. First, bacteria use various catabolic pathways for biosynthesis. Glycolysis, pyruvate oxidation, and the TCA cycle are bidirectional pathways and are considered central amphibolic pathways. 2. By using the glyoxylate bypass of the TCA cycle, bacteria can use acetate not as an energy source but rather as a precursor of a variety of 4 carbon dicarboxylic acids. 3. Amino acid biosynthesis falls into convenient groupings based on points of origin in the central pathways. A. In the glutamate family, glutamate and glutamine arise from reductive amination of alpha-ketoglutarate and evolve to provide the carbon skeleton for proline and arginine. B. In the aspartate family, aspartate is synthesized by transamination of oxaloacetate, gives rise to asparagine, and can be reduced to lysine, methionine, threonine, and isoleucine. C. The pyruvate family of alanine, valine, isoleucine, and leucine results from the initial transamination of pyruvate. D. The serine family uses serine, glycine, and cysteine is derived from a 3P glycerate with glycine resulting from transfer of the 1C fragment from serine to tetrahydrofolate. E. Histidine is synthesized from the 5C backbone of phosphoribosyl pyrophosphate PRPP and is unique in that its carboxyl group is not present in the starting compound but is formed later. F. The aromatic amino acids have shikimic acid as a common precursor. Tyrosine, phenylalanine, and tryptophan are eventually derived from this common pathway. 4. Nucleotide biosynthesis in bacteria is identical to that in animal tissue. Purine nucleotides are built on the ribose P chain from PRPP, glycine and carbon and nitrogen additions in a long sequence. B. Pyrimidine nucleotides are formed via a series of carboxyl containing intermediates starting with carbamyl phosphate with ribose P added late in the sequence. 5. The principal macromolecules DNA, RNA and protein are polymers of the nucleotide and amino acid building blocks and are synthesized via a highly regulated system in the cell cytoplasm. A. DNA acts as a template necessary for its own synthesis as well as for that of RNA. B. RNA serves as a template for protein synthesis and provides the core of ribosomes on which the proteins are synthesized. C. The sequence of information transfer from a macromolecular synthesis involves replication, DNA to DNA, transcription, DNA to RNA, and translation, RNA to protein. Chapter 3, Bacterial Genetics. First, organization and replication of the genetic information. A. The chromosome in living bacterial cells is a single continuous strand of DNA. First, the average bacterial chromosome has a molecular weight of 3 × 10 to the 9th and is a closed circular structure. 2. The DNA is a native double-stranded form with the usual AT and GC base pairing. B. Genes. Genetic information encoded in the bacterial DNA is organized into a series of units known as genes. First, the normal bacterial chromosome encodes only one copy of each gene. Bacteria are therefore haploid organisms. 2. A gene is a chain of nucleotides, the transcript of which is read without interruption and results in a polypeptide chain. 3. The genetic information in a gene is encoded in triple nucleotide groups, codons, each triple group coding for one specific amino acid. 4. Genes in eukaryotic organisms usually have intervening sequences, introns, which are removed, sliced or spliced out during messenger RNA, mRNA processing. However, bacterial genes are contagious. 5. Genes are often defined functionally by a phenotype, and their boundaries are identified by genetic rather than biochemical tests. A. Fine structure genetic mapping involves the determination of the recombination frequency and may be used to locate mutations within a single gene. B. By the construction of partial diploids with one well-defined gene copy complementation testing may be used to define gene boundaries. C. Replication of the bacterial chromosome is a precise process ensuring that each daughter cell receives an exact copy. First, bacterial DNA replication begins at a specific site of on the chromosome, the origin of replication. A. Local denaturation of the 2 DNA strands occurs. B. Each of the separated strands serves as a template for a complete round of synthesis. 2. Each daughter cell receives one parental strand and one newly synthesized strand of DNA, a process termed semi-conservative replication. 3. Initiation of chromosome replication is carefully controlled to coordinate with cell division. A. Bacterial protein synthesis is required for the initiation of DNA replication. B. Rapidly growing cells often average more than a single chromosome per cell. C. Bacterial cell division is usually coupled to the completion of a full round of DNA replication. Second, gene expression and regulation. Gene expression and regulation. Bacterial genes are precisely regulated to allow for balanced growth under a wide variety of nutritional conditions. A. Several regulatory mechanisms work at the metabolic level and have little effect on the expression of the genetic information. First, end product inhibition. Feedback inhibition is a mechanism that causes the end product of a pathway to inhibit the first enzyme of the pathway pathway directly. A. End product inhibition is usually direct and immediate. B. It usually exhibits competitive antagonism between the pathway end product and the enzyme substrate. First, this competition is usually not for a single binding site but involves alternative forms of the enzyme. Second, enzymes that demonstrate alternative forms as a function of interaction with a variety of regulatory molecules are designated allosteric. 2. Enzyme activation is a mechanism that causes positive effector molecules to stimulate the first enzyme of a pathway. B. Initiation of transcription. Regulation at this level usually affects a small group of genes involved in a process, an operon. First, enzyme induction involves the synthesis of new mRNA and a new polypeptide in response to the presence of a specific type of molecule, an inducer, which is not necessarily a substrate for the enzyme. Second, end product repression functions in reverse of enzyme induction through the decrease in enzyme formation by the end product of a pathway. 3. Both inducible enzyme systems and end product repressible systems are under negative control, mediated by an active repressor or aporepressor molecule. A. In inducible enzyme systems, the repressor is active in its native configuration. B. In end product repression, the repressor alone is inactive but is converted to an active form after binding the end product effector, a co-repressor. 4. The operator is defined as the genetic site of action of the repressor molecules. A. A group of genes controlled by a single operator constitutes an operon. B. Mutations in the operator may lead to the inability to bind the repressor, thereby giving a non-repressible phenotype, the OC mutation. C. Operons begin with a promoter, P, which is the site of RNA polymerase binding, and are followed by the operator, which is followed by the structural genes. 5. Repressor molecules are proteins that recognize specific DNA sequences, the operator, and are also able to bind the inducer or a co-repressor. A. The LAC repressor is a 150,000 molecular weight polypeptide of 4 identical subunits, and it binds to double-stranded but not single-stranded DNA. B. Repressors bind DNA with extremely high affinity, and therefore only a few molecules per cell are required. 6. Catabolite repression, gene expression and regulation continuation. 6. Catabolite repression by glucose has a broader effect than that observed with specific end products. A. The glucose effect involves an interaction with the promoter rather than with the operator. B. The signal molecule in the repression is 3,5 cyclic adenosine monophosphate cyclic AMP, CAMP, which acts in concert with the CAMP repressor protein, C-reactive protein, CRP, or CAMP binding protein, CAP. 7. Transcription of bacterial DNA is mediated by RNA polymerase in connection with additional factors. A. Purified RNA polymerase is found in 2 forms, core enzyme and holoenzyme. B. The holoenzyme contains an initiation factor, sigma factor, which promotes the attachment of the enzyme to specific initiation sites. C. Initiation of transcription involves the interaction of the sigma factor containing enzyme with the promoter at a well-defined binding site of approximately 40 base pairs in length. D. Following initiation, chain elongation proceeds through the addition of single mononucleotides. E. Termination of transcription occurs at specific DNA sites where the stability of the DNA helix is altered. For instance, a GC-rich which region. C. Transcription, termination, and translation. Regulation of gene expression also occurs at these levels. First, two mechanisms control gene expression at the level of transcription termination. A. Polarity occurs in operones that are transcribed as a single mRNA species, and it works by transcription termination at weak terminator sites within the operone, leading to higher levels of mRNA for proximal genes of the operone than for distal genes. B. In some operones there is a variable premature termination of transcription in the early mRNA known as the leader sequence, called attenuation. 2. Although of minor significance in the overall regulation of gene expression, several examples of regulation of translation have been observed. Third, the molecular basis of mutation. The molecular basis of mutation. Mutation refers to all heritable changes in nucleotide sequence arising within an organism, whether induced or spontaneous. A. DNA changes are associated with many mutations and have been surmised from genes of which the precise base sequence and the sequences of their protein product are known. These changes fall into three broad categories. First, nucleotide replacements are simple changes of one nucleotide base for another. A. Transitions are those replacements in which one purine is replaced by another, or one pyrimidine is replaced by another, for instance AT to GC. B. Transversions are those replacements in which one purine is replaced by a pyrimidine, or one pyrimidine is replaced by a purine, for instance AT to CG. 2. Losses of nucleotides result in deletion, which may be of single bases or very large nucleotide sequences. A. Microdeletions of a single nucleotide generate mutation by shifting the triplet code, reading frame, and totally change the amino acid sequence of the protein downstream of the mutation. B. Large deletions may or may not change the reading frame. However, they do lead to the loss of the coding capacity for only part of a gene, or for as many as several genes. 3. Nucleotide insertions are usually of a single base pair and result in a change of the reading frame similar to that caused by a microdeletion. B. Point mutations are nucleotide replacements and single nucleotide insertions and deletions that, through appropriate replacement, revert to a wild-type combination. First, point mutations will give wild-type recombinants with each other as well. 2. Large deletions result in wild-type recombination only when the entire deleted portion is restored and will not revert. 3. Nonsense mutations are produced when a single nucleotide change results in the introduction of a termination codon in place of an amino acid codon. 4. Missense mutations are produced when a single nucleotide change results in the substitution of one amino acid codon for another. A. Altered enzymes resulting from a single amino acid change often exhibit greater thermal liability and are designated temperature sensitive. B. Even when a missense mutation leads to the complete loss of enzymatic function, the polypeptide produced usually retains its antigenic character, and the cells produce an immunologically cross-reacting material, CRM. C. Mutagens are agents that are active in DNA damage, which leads to mutation. First, radiation, both ionizing and non-ionizing, is a common source of mutagenesis. A. Most mutations arising from ultraviolet radiation are caused during the post-replication repair of ultraviolet-induced DNA damage. B. Mutations induced by ionizing ionizing radiation, which leads to strand breaks, also appear to rise following inefficient post-replication repair. 2. Chemical mutagens, which are the most common inducers of mutations, fall into several main classes. A. Base analogues are incorporated into DNA during normal replication, but because they tend to undergo transient internal rearrangements, tautomerization, they are copied incorrectly in the next round of replication. B. Deaminating agents such as nitrous acid and hydroxylamine induce high-frequency selective transitions. C. Alkylating mutagens such as the strongly reactive nitrose guanidine or the mono-functioning alkylating agents like ethyl ethane sulfonate also produce transition mutations. D. Acridine derivatives can intercalate successive base repairs of DNA and usually cause reading frame shift mutations. D. Restoration of enzymatic activity in a mutant lacking enzyme activity usually occurs via one of two different mechanisms. First, true reversion is a change at the site of the original mutation that restores the original gene function. 2. Suppression of a mutation is the phenotypic restoration of gene function by a change at a site different from the mutation. 4. Genetic recombination and gene transfer. Bacteria are haploid organisms that use a series of primitive mechanisms or gene transfer. All of these mechanisms produce partial diploids, merozygotes, as a prelude to a recombinational event. A. Transformation was the earliest recognized form of bacterial gene transfer. It involved the conversion of a non-encapsulated strain of pneumococcus R cells to an encapsulated strain S cells. First, transformation has been observed in cultures of a wide variety of both gram-positive and gram-negative genera. Second, the active component in transformation is naked DNA. A. The uptake of DNA by cells requires energy together with a convenient site of entry, a combination termed competence. B. Some bacteria immediately convert the double-stranded DNA to single strands following uptake, whereas others leave the DNA in a double-stranded form prior to recombination. 3. Some species of bacteria that are not normally transformed can be induced to become competent by treatment with calcium chloride, CaCl, and low temperature. 4. Transformation is a genetic tool that can be used for gene mapping and the quantitative determination of gene dosages. 5. In nature, transformation probably occurs most frequently between members of the same species. B. Transduction, like transformation, introduces only a fractional portion of the entire bacterial chromosome into the recipient cell. First, transduction involves the introduction of DNA by a bacteriophage. A. Because the DNA is packaged into bacteriophage code, transduction is easier to perform than transformation. B. Transduction is more reproducible than transformation. 2. Two mechanisms of bacterial transduction have been described: generalized and specialized. A. In generalized transduction, the phage packages host DNA at random and therefore may transfer any gene. B. In specialized transduction, phage DNA has been integrated into the host chromosome, incorporates a few specific genes upon excision, and is limited to the transfer of those genes. 3. The generalized transducing particle carries only a fragment of the bacterial chromosome and has no bacteriophage DNA. Because the generalized transducing particles are limited in the size of the DNA fragment that they can package, they are very valuable as genetic mapping tools. 4. Specialized transducing phages are only a very small proportion of the progeny from a normal lysogenic culture and always contain both phage and bacterial genes. Specialized transduction is generally limited to those genes near the attachment site of available temperate phages. A. Lysates from normal lysogenic cultures cause low-frequency transduction, LFT. B. High-frequency transduction, HFT. Lysates are rich in transducing particles and can be produced with by propagating the phage from cells transduced by LFT. First, with non-defective transducing phages, the particles in a single plug are sufficient. Second, with defective transducing phages, the cells must be co-infected with a normal helper phage and are therefore double lysogenic. C. Conjunction in bacteria, unlike the other two mechanisms of gene transfer, demonstrate a marked polarity. First, the fertility F factor, which is also known as the sex factor, codes for a series of genes necessary for conjugal transfer. Escherichia coli may exist in one of two heritable mating types, donor F or recipient. Donor is F plus or recipient F minus. A. In a given genetic cross, only the recipients must be viable. The donors need only to be able to transfer their DNA. B. The critical factor in the recognition and contact between the donor and the recipient is the F pilus, which is coded for by the F factor. C. The DNA transfer between F plus and F minus organisms occurs through a bridge between the two organisms. First, cell-to-cell contact is necessary in addition to contact by the pilus. Second, what direct role, if any, the pilus plays in DNA transfer is unknown. Second, the donor property of F plus cells is the result of the presence of a plasmid DNA, which is the F factor. A. The F factor was the first known conjunctive plasmid. B. The F factor transfers itself at very high frequency, quickly converting an F minus population to F plus. 3. During conjugation, the donor cell transfers only part of a chromosome to the recipient. A. The extent of chromosome transfer is dependent on the duration of mating. However, total chromosome transfer is very rare, even when adequate time is given. B. One of the most powerful genetic tools in conjugation is mapping by interrupted mating. 4. The F factor must be incorporated into the bacterial chromosome in order to mobilize its transfer. A. Strains in which the F factor is integrated stably into the bacterial chromosome transfer bacterial DNA at 1,000 times the frequency of simple F plus cultures. B. Cultures with the F factor stably integrated are termed high frequency recombination HFR strains. C. At low frequency, the F factor is excised from the HFR chromosome, and on rare occasions it may take a small segment of the bacterial chromosome with it. First, these F factors containing a small chromosomal segment are termed F prime factors. The F prime factors transfer with the extremely high frequency of the F factor alone and are very useful in achieving an extremely high frequency transfer of selected genes. 5. Independently isolated HFR strains differ in the sequence of gene transfer. However, the gene sequence determined from a series of transfers is circularly permuted. A. The genetic map of E. coli is a closed circle. B. The different HFR isolates may transfer in opposite directions depending on the orientation of the integrated F factor. C. Transfer of the entire circular genetic map of E. coli takes 90 to 100 minutes, and the chromosome is conveniently presented as a closed circle of 100 equal parts, 100 map units. D. Plasmids such as the F factor act on a group of independent extrachromosomal genetic elements. First, plasmids fall into two classes: conjunctive like the F and resistance plasmid R factors, and non-conjunctive like the small bacteriocin factors. A. The conjunctive plasmids are usually quite large, 60 to 120 kilobases, and are maintained at 1 to 2 copies per cell. The conjunctive plasmid have two main functions, that of replication and transfer, and that of the other material carried on the plasmid. First, the conjunction and replication functions may associate with other genes, such as those for bacteriocin or toxin production. Second, R factors are of great medical interest since they may confer resistance to many types of drugs. A. Widespread antibiotic therapy together with interspecific transfers has selected a large number of multiply resistant strains in several species. B. The drug-resistant R genes are often part of a highly mobile transponsus, discrete genetic and physical entities that are able to move from one position to another in a genetic and physical entity that are able to move from one position to another in a cell and likewise move to a new genome. These transposons assort in random fashion to produce the wide variety of R factors encountered in nature. C. R genes usually code for specific enzymes that modify or destroy specific antibiotics. B. The non-conjunctive plasmids are usually small, 1.5 to 15 kilobases, and are present in multiple copies, 10 to 20 per cell. Non-conjunctive plasmids lack the functions required to mobilize their own transfer, although they may be mobilized, transferred by conjunctive plasmids. Second, plasmids have the ability to recombine with each other and lead the bacterial chromosome. A. Recombination occurs between segments of chromosome and segments in the plasmid. The insertion sequences ISS, which are specific nucleotide sequences that range from 800 to 1,400 base pairs in length. B. ISS are found as parts of transposable elements, transposons. E. Genetic recombination at the molecular level involves similar mechanisms regardless of the DNA. First, generalized recombination involves the alignment of similar base sequences from two different sources. A. In symmetric assimilation, single DNA strands from each of two sources are incorporated into the other in the form of DNA heteroduplex. B. In asymmetric assimilation, a single DNA strand from only one part is involved in the transfer. 2. Generalized recombination in E. coli involves the partition of at least 3 gene products, although other genetic loci are also participate to a lesser extent. A. The REC-A gene is involved in both recombinations and repair processes, and its absence leads to an almost complete loss of generalized recombination. B. The REC-B and REC-C genes code for the two polypeptides of an adenosine triphosphate ATP-dependent nuclease, exonuclease 5. C. The loss of the REC-B and REC-C exonuclease does not decrease recombination to the same extent as does the loss of the REC-A function. 3. Site-specific and illegitimate recombination occurs by a process independent of the REC-A gene product. A. Site-specific recombination occurs between two identically matched sequences with the intervention of a gene product specific to that recombination. B. The most common examples of this type of recombination are the integration of bacteriophage lambda at the at site and the insertion of transposable genetic elements. 5. Genetic manipulation of bacteria. In the recent past, novel techniques for rearranging genes with bacteria and molecular recombination between DNAs of any source revolutionized genetics. A. Operon fusion is one approach to the study of gene regulation and operation activity. First, fused operones are transcribed as a single unit under the control of the normal components that control the operator. Second, operone fusions rely on the selection of deletion mutations which remove the regulatory elements of one operone, thereby connecting it to the terminus of another. B. Molecular cloning through recombinant DNA technology permits a segment of DNA from any source to be inserted into a bacterial replicon, a plasmid or bacteriophage, and replicated in a bacterial host. First, a wide variety of restriction endonucleases cleave DNA at highly specific sites and leave overlapping complementary ends, which after re-annealing can be closed with polynucleotide ligase. Second, when these cleaved DNA molecules are mixed with plasmid DNA, similarly cleaved, occasionally a piece of foreign DNA re-anneals with the plasmid to form a recombinant chimeric plasmid. 3. Chimeric plasmids may be introduced into E. coli by transformation, and the plasmids will be propagated, cloned in the cell's progeny. 4. The types of bacterial replicons have been used. Two types have been used for the cloning foreign DNA. A. Mutants of L-Phag, of lambda phag, which allow for rapid screening of specific DNA inserts, have been derived. B. Varieties of small non-conjunctive plasmids that carry one or more antibiotic resistance markers and are particularly valuable in the production of cloned gene translation products. 5. Despite the ready replication of eukaryotic DNA in E. coli, the organization of eukaryotic DNA and the nature of initiation signals are clearly a barrier to proper expression of eukaryotic genes in prokaryotes. Chapter 4. Antimicrobial agents. First, methods of microbial control. The inhibition of growth and the destruction of pathogenic microorganisms are accomplished by either physical or chemical means. Some methods of microbial control are applied only to inanimate objects, fomites, while other methods display selective toxicity and may be used in vivo. A. Physical methods. The reduction in numbers, for instance disinfection and total elimination, that is sterilization of potentially pathogenic microorganisms from the environment, usually are accomplished by physical methods. First, heat is employed in a variety of methods for reducing or eliminating microorganisms from heat-stable materials. A. Pasteurization is the destruction of pathogenic microorganisms by rapid heating of a substance to 71.7 degrees Celsius for 15 seconds, followed by rapid cooling. Pasteurization does not sterilize the substance, since all microorganisms are not susceptible to this treatment. This technique has been successful in eliminating foodborne disease such as gastrointestinal tuberculosis and Q fever. B. Dry heat of 160 degrees Celsius sterilizes a material exposed to this temperature for 2 hours. This exposure time is necessary to kill all microbial spores, although vegetative cells are destroyed within the first few minutes. Dry heat sterilization chars organic compounds and causes excessive evaporation of liquid materials. C. Moist heat, that is steam of 121 degrees Celsius under a pressure of 15 pounds per square inch, is the most effective means of sterilizing heat-tolerant liquids. Heat-resistant spores are killed in less than 15 minutes in small volumes of liquid. Longer periods of time are required to equilibrate volumes of liquid over 500 ml to the sterilization condition. An autoclave which essentially is an industrial pressure cooker is used for moist heat sterilization. Second, radiation of varying wavelengths in the electromagnetic spectrum is used for disinfection and sterilization of heat labile materials. A. Ultraviolet UV light between the wavelengths of 250 nanometers and 270 nanometers is absorbed by nucleic acids. UV light damages microbial cells by disrupting hydrogen bonds and causing the formation of thymine dimers and DNA. This structural alteration of the DNA often results in lethal frame shift mutations. UV light has limited application for sterilization because of its poor penetrating energy and its absorption by glass and water. B. Gamma radiation and X-rays are forms of ionizing radiation that effectively sterilize many materials but must be used cautiously because of their potential danger to human cells. These forms of radiation cause the formation of free radicals which chemically react with proteins and nucleic acids, resulting in cell death. Gamma radiation is used extensively for sterilization of plastic materials and is receiving renewed interest in the United States as a means of preserving foods. C. Microwaves have been used in the microbiology laboratory for rapid re-sterilization of media that has been stored for extended periods of time. However, sterilization is the result of the heat produced by the radiation rather than by direct effect of microwaves on microbial cells. 3. Filtration of liquids and gases through filters made by natural or synthetic materials is an effective means of removing bacteria and eukaryotic microorganisms. Membrane filters with the pore size of 0.2 micron effectively remove all bacteria but do not actually sterilize the liquid or gas because most viruses are not retained by these filters. A practical limitation of filtration is that the increasing viscosity of the liquid being filtered and the small pore size of the filters decrease the flow rate. B. Chemical methods of microbial control include disinfectants and antiseptics, which are non-specific for the cells that they affect, and antibiotics and synthetic antimicrobial agents which have a selective toxicity. First, non-selective chemicals that control the growth of microorganisms on inanimate objects are referred to as disinfectants, while those applied to human tissue are known as antiseptics. These chemicals ideally should be effective killers to all microorganisms, including viruses, should be soluble in water or for ease of preparation and application, should have a low toxicity for humans, and should be reasonably economic. A. Alcohols such as ethanol, isopropanol, and benzyl alcohol are effective antiseptics when used as 50 to 70% aqueous solutions. Alcohol precipitate proteins and solubilize lipids present in cell membranes. As solvents, they also effectively clean human tissue. Alcohols kill the vegetative cells of many bacteria when properly used, but they do not affect microbial spores, fungi, and most viruses. B. Halogens, particularly iodine and chlorine, are widely employed as antiseptics and disinfectants. Iodine is solubilized in ethanol is a common antiseptic for cuts and abrasions. The iodine reacts with hydroxyl groups and inactivates proteins. Chlorine gas reacts with water to form hypochlorous acid, which in turn reacts with water to form hydrochloric acid and hydrogen peroxide. Both of these compounds are strong oxidants and kill microbial cells. Household bleach, 5.25% sodium hypochlorite, is another source of hypochlorous acid and is an effective disinfectant. C. Aldehydes such as formaldehyde and glutaraldehyde are alkylating agents that react with the amine, sulfhydryl, and carboxyl groups of proteins proteins and small organic molecules to kill microorganisms. Formaldehyde 8% and glutaraldehyde 2% are effective disinfectants but have limited use because of their noxious vapors. D. Heavy metals are effective as antimicrobial substances because of their ability to precipitate proteins and other organic molecules. Silver nitrate, copper sulfate, and merbromine mercurochrome are widely used as antiseptics. Lead, arsenic, and inorganic mercury rarely are employed as disinfectants because they are concentrated by human tissue and cause cell death. E. Phenols and their substituted derivatives are highly effective disinfectants, and lower concentrations are used as antiseptics. Phenols function by denaturating proteins and disrupting cell membranes. Phenol no longer is used widely, but phenolic derivatives such as carbolic acid and lysol are common disinfectants. Hexachlorophane is an excellent anti-staphylococcal antiseptic available by prescription. F. Cationic detergents contain alkyl groups that interact with membrane lipids and cause the disruption of the cytoplasmic membrane of bacteria. Quaternary ammonium compounds are bactericidal but have a low toxicity for mammalian cells and can be used as effective antiseptics. Cationic detergents are inactivated by low pH phospholipids, organic compounds, and metal ions. G. Gases of various types have been employed as disinfectants since ancient times. Sulfur dioxide is used as a food preservative. Ethylene oxide and propylene oxide are effective sporicides when used under pressure for the sterilization of plastic materials. 2. Selective chemicals that are used for their ability to inhibit the growth of or kill microorganisms include naturally occurring antibiotics, semi-synthetic antibiotics, and some synthetic chemical compounds. Many of these agents are used in the treatment of infectious diseases because of their low toxicity for mammalian cells. A. Antibiotics are natural substances produced by an organism to kill or inhibit the growth of another organism. Not all antibiotics are effective therapeutic agents because of their toxicity for human cells. Semi-synthetic antibiotics generally are synthetic derivatives of naturally occurring antibiotics. B. Chemotherapeutic agents are chemicals that are used for treating infectious diseases. Frequently, these agents are analogues of microbial cell constituents or substrates. C. The efficacy of a drug is the result of its qualities that make it useful in the treatment of infectious disease. These qualities include stability in vivo, rate of absorption, rate of elimination, and the ability to penetrate the infected site. D. The therapeutic index of a drug is the minimal dose causing toxicity to the host divided by the minimal dose required for antimicrobial activity. Therefore, the higher the ratio, the greater the efficacy of the drug. E. The attainable serum level of a drug is dependent on the dosage of a drug administered, the host's body weight, the route and schedule of administration of the drug, and the rate of elimination. 2. Characteristics of the major classes of antimicrobial agents. The major groups of antimicrobial agents can be classified on the basis of their site of action on the microbial cell. Most clinically employed antimicrobial agents are inhibitory only at normally achievable concentrations and do not kill the microbial cells. In addition, this inhibitory effect usually is exerted only on actively growing microorganisms. A. Inhibitors of cell wall synthesis. First, penicillins are produced either naturally by a species of the fungus Penicillium or by synthetic modification of penicillinic acid. The penicillins primarily are effective against gram-positive bacteria and a limited number of gram-negative bacteria. These agents may act as structural analogues of D-alanyl D-alanine and inhibit the enzymatic reaction responsible of the terminal peptide link linkage between alanine and glycine during the synthesis of the murine component of bacterial cell walls. The result is that the cells are vulnerable to osmotic pressure and lyse, although under special conditions the bacteria may survive as L forms with incomplete cell walls. Penicillin G, the first antibiotic effective in systemic bacterial diseases, has the disadvantage of being inactivated by beta-lactamase produced by bacteria and the acid pH of the stomach. Chemical modification of the molecule has produced semi-synthetic penicillins which overcome these disadvantages to varying degrees. These agents include penicillin V, ampicillin, oxacillin, methicillin, and numerous others. The penicillins can be administered orally and have a low toxicity for mammalian cells, although a few individuals are allergic to these compounds. 2. Cephalosporins are produced by species of the fungus Acremonium, formerly called Cephalosporium. They are structurally similar to the penicillins in that they contain the beta-lactam ring. The original naturally occurring compounds have been chemically modified to produce a large number of cephalosporin antibiotics. So-called third-generation cephalosporins are available currently for clinical use. These agents inhibit cell wall synthesis in a broad spectrum of gram-positive and gram-negative bacteria. The beta-lactam ring of the cephalosporins is more protected than it is in the penicillins and is less susceptible to the action of beta-lactamase. Certain bacteria, however, produce cephalosporinase and are resistant to the antibiotics. Cephalosporins can be given to individuals with allergies to penicillins. 3. Bacitracin is a peptide antibiotic produced by Bacillus subtilis. The antibiotic is produced just prior to sporulation of the bacilli by an enzymatic process rather than by a normal process involving transfer of ribonucleic acid and ribosomes. The antibiotic prevents peptidoglycan synthesis. It is relatively toxic for humans, but it is absorbed poorly, making it useful as a topical agent for the treatment of infected wounds. 4. Vancomycin is produced by species of Streptomyces and prevents the formation of the peptide portion of the peptidoglycan molecule. It is effective against gram-positive bacteria. It is a high molecular weight antibiotic, and the chemical structure has not been confined. 5. Cycloserine also is produced by a Streptomyces species. It inhibits cell wall synthesis by interfering with the production of alanine and its incorporation into the interpeptide bridges of peptidoglycan. B. Antibiotics acting on cell membranes. Antibiotics acting on cell membranes. First, polymyxin is produced by Bacillus polymyxa and inhibits the normal function of the bacterial cell membrane. The polymyxins act as cationic detergents to disrupt the bacterial cell membrane. These antibiotics are too toxic for internal use, and only polymyxin B and polymyxin E colistin are used clinically as topical antibacterial agents. Second, the polyane antibiotics nystatin and amphotericin B, which are produced by Streptomyces species, are clinically useful as antifungal compounds. The polyanes bind to naturally occurring ergosterol molecules in the fungal cell membrane, resulting in the leakage of small molecules out of the cells. Nystatin currently is used as a topical antifungal agent, principally in treating mucocutaneous candidiasis. Amphotericin B is used in the treatment of systemic mycosis, but it is highly nephrotoxic because it also binds to cholesterol in mammalian cell membranes. 3. The imidazoles can cause leakage of molecules through the cell membrane of fungi. It is believed that the imidazoles inhibit a metabolic step that is important in the synthesis of ergosterol. These compounds have a broad broad-spectrum antifungal action and also are inhibitory to some bacteria and protozoa. Clotrimazole is limited to topical use in the United States. Miconazole is administered parenterally, and ketoconazole is effective as an orally administered compound. C. Inhibitors of protein synthesis. First, the aminoglycosides inhibit protein synthesis in bacteria by binding to the ribosome. The mode of action of streptomycin is known in more detail than that of other aminoglycosides. Other aminoglycosides such as amikacin, kanamycin, neomycin, gentamicin, tobramycin probably act by a similar mode of action. Streptomycin binds to the 30S subunit of the bacterial ribosome by irreversibly binding to a specific structural protein of the subunit. This binding produces three effects on protein synthesis. A. It blocks normal function of the initiation complex. Streptomycin ribosome complexes prematurely leave the messenger RNA assembly line. These ribosomes subsequently dissociate into their 30S and 50S subunits and reassemble at the normal initiation site on mRNA. However, they remain irreversibly inactivated initiation complexes that cannot form peptide bonds. This is the most important mechanism of the bactericidal activity of streptomycin. B. It interferes with the attachment of transfer RNA, tRNA, to the ribosome mRNA complex. It causes the production of faulty proteins by distorting the triplet code of mRNA and causing misreading. The misreading results in the insertion of the wrong amino acids into the polypeptide chain. Second, the tetracyclines are broad-spectrum antibiotics produced by Streptomyces species that have a hydronaphthasine nucleus consisting of 4 rings. Chlortetracycline, oxytetracycline, doxycycline, and related compounds bind to the 30S subunit of bacterial ribosomes. The attachment of tRNA to the ribosome mRNA complex is blocked, and this prevents the introduction of new amino acids into the polypeptide chain. 3. Chloramphenicol is the only naturally occurring antibiotic that contains nitrobenzene, and it probably is this chemical structure that accounts for its toxicity to bacterial and mammalian cells. The drug combines with the 50S subunit of bacterial ribosomes. It prevents peptide bond formation by blocking the action of peptidyl transferase, which is located in the 50S subunit. Anaplastic anemia is a serious side effect in patients receiving chloramphenicol therapy. 4. Erythromycin is produced by Streptomyces erythraeus, and it is the only clinically important member of the macrolide antibiotics. The basic chemical structure of a large lactone ring to which unusual sugar molecules are attached. The mode of action is analogous to that of chloramphenicol. The molecules bind to the 50S subunits of ribosomes and interfere with peptidyl transferase activity. 5. Lincomycin is similar to erythromycin in its antibacterial activity and in its mode of action. However, chemically it is dissimilar, consisting of an amino acid attached to a sulfur-containing amino sugar. The drug prevents peptide bond formation by binding to the 50S subunit and blocking peptidyl transferase activity. Interestingly, all Escherichia coli are resistant to the growth-inhibiting action of lincomycin. Clindamycin, that is 7-chloro-7-deoxy-lincomycin, is a synthetic modification of lincomycin used in the treatment of infections caused by anaerobic bacteria. 6. Emetine is an ancient antimicrobial compound that is the principal agent of the anti-protozoan compound ipecac. It inhibits the transfer of amino acids from tRNA to the polypeptide chain of the ribosome, preventing elongation. The drug is used in the treatment of amoebic dysentery and liver abscesses, but it is relatively toxic to the patient, probably because emetine also inhibits protein synthesis in mammalian cells. 7. Thiosemicarbazones are synthetic compounds that appear to interfere with protein synthesis in smallpox virus by disrupting polyribosomes. This effect is produced by the ability of thiosemicarbazones to break mRNA into small fragments that prevent polyribosome formation. D. Inhibitors of transcription and nucleic acid synthesis. First, rifamycin antibiotics are products of fermentation reactions by Streptomyces mediterranei, although the currently most important compound, rifampin, is a semi-synthetic derivative of rifamycin B. Rifampin consists of a double ring structure with a long aliphatic bridge and a nitrogenous side chain. The drug is a potent inhibitor of DNA-dependent RNA polymerase